for coming. We will be getting started here shortly. We're allowing people time to get into the room. And if you would be so kind for the presenter to turn on your camera so we can see your beautiful faces, that will be great. Give us about 2 minutes and we'll let people in and we'll get started, okay? Thank you. Good afternoon, everyone. My name is Dr. Trey Waller, and I work in the SEED office, which is the Center for the Enhancement of Engineering Diversity, and this is Master Your Future. We're excited to have with us Dr. Jonathan Auguste. He is an assistant professor here at Virginia Tech, and he is going to be speaking to us about striving for excellence so we can create opportunities for others. We're excited to have you, Dr. Jonathan. The floor is yours, and we look forward to hearing your presentation.
Thank you, Trey. I appreciate the nice introduction. It is truly a pleasure to speak with everyone here today and to be able to present a little bit of my research as well as some encouragement for everyone today. So as Trey mentioned, my name is Jonathan. I do actually go by Jonathan, so if you do see me, please feel free to call me Jonathan. One quick disclaimer, the points that I raised today represent my viewpoints. I may not necessarily reflect the stance of my institution. All right. So I run a robust arboviral research program out of Fralin Hall in the Department of Entomology. And I'm going to tell you a little bit about the research that we do in our lab, and as well as introduce you to some of the folks in my lab. So we have a very, very diverse research portfolio ranging from vaccine development for arthropod-borne viruses like flaviviruses and alphaviruses, as well as viruses like SARS-CoV-2. We also do a lot of viral pathogenesis studies. We do a lot of preclinical animal model development. And here we're actually able to work with a large, diverse array of animals. We work with chickens, quail, ducks. We work with mice. We're going to start working with guinea pigs, and we do a lot of field work with wild bird species as well. My lab also does a lot of virus ecology work, where we do primarily surveillance programs as well as experimental infections of both vertebrate animals as well as arthropod hosts. So we do love to go out in the field and collect our mosquitoes and birds. We also do some virus-host interaction studies where we look at how viruses function together in a synergistic or in a competitive way inside host cells to either exclude other viruses or to perform well in combination with other viruses. And here I have a photo of 4 of the graduate students in my lab and my lab manager, and I'll introduce you to them a little later on in the story. But today, I'm very happy to have the pleasure to tell you my story. And I wanted to start us off by saying one quick quote. Life gives us experiences for personal development. Appreciate the lessons and be a learner. And that's one thing that I want to really reiterate today. Always be a learner. That's the nature of life. And as we learn more, we achieve more. So my story actually starts in the Caribbean island of Trinidad and Tobago. It's where I was born, where I was raised, where I did my PhD, where I did my bachelor's degree. Growing up, my family was slightly competitive, but we were primarily into athletics and sports. So I grew up at a slightly competitive household, but it was a very supportive environment. Upon pursuing my Bachelor's of Science at the University of the West Indies, I pursued a major in biochemistry. And because of my athletic background, I really wasn't the top or the best student. I was much more interested in pursuing athletics. But then one faculty member in particular, Dr. Brian Coburn, he realized what was going on. He realized that I wasn't being challenged in any way, even though I was pursuing a pretty difficult major. And this guy is the one who started that challenging process when I was in undergrad. And it really, really changed the way I viewed my academic standing. And it really, really did push me forward a lot. Now, even though I did get challenged, I started a little too late. I graduated with a GPA of about 2.99, so I didn't even make a 3.0. And if you're from the Caribbean, you know, grades mean a lot. You would not get in a graduate school with a GPA of less than 3. However, Dr. Coburn was very nice, and he wrote a really nice letter of support for me to Dr. Christine Carrington, who went out on a limb, took a chance. Again, usually we would not accept students as a GPA less than 3. And she recruited me as a PhD student. And that worked out well. She was a remarkable mentor, and I had more than 8 publications at an institution where a publication of n equal 1 was being successful. So we had a really good research experience and a really good mentorship experience during both my bachelor's and PhD experience. Next, I want to tell you just a little bit about some of the research that I did in Trinidad and that what we do here with respect to the virus ecology component. So as I mentioned before, we do a lot of surveillance. This first started while I was doing my PhD. Trinidad has a long history of arbovirus research. Back in the 1950s and 60s, there was a lot of work done on arbovirology in Trinidad. There, they isolated hundreds of viruses, many of which were completely novel and new viruses. They serologically classified the viruses, they taxonomically identified the vectors, and they did a lot of studies looking at virus and vector ecology. However, since the 1950s and 60s, Trinidad has changed considerably. Back then, sugarcane was our primary export, but now we export oil. So in recent times, we have increased rates of urbanization. We have a lot of deforestation of our pristine habitats for new housing developments for our growing population. We have a lot of squatting or humans encroaching on forested areas. And we have a lot of travel through Trinidad to the rest of South America as well. Well, my question back then was like, how does this urbanization or these anthropogenic changes impacting the virus and vector populations in Trinidad? So to do that, we set up a very simple mosquito surveillance study. We did surveillance for 2 years at 2 sites, the Arepa savannas and the bush forest. We set a variety of different mosquito traps, but we collected about at each site almost once a week. And we did this at different elevations. We also, for the love of science, we also did a lot of opportunistic human sampling. I don't think this is approved, but back then the easiest way to isolate a virus was to get infected with it. So we would go there and we get bitten by mosquitoes and try to catch them as they bite us. Eventually, when we do collect these mosquitoes, we would take them back to the lab and we would sort these mosquitoes into pools for which we use for our screening. So within 2 years, we collected 185,000 mosquitoes. That was a remarkable number for a small team of about 2 to 3 people. We sorted these mosquitoes by species into 9,000 pools, representing more than 46 species. Of the more than 46 species that we detected, approximately 32 were known or incriminated vectors. But by far, we had 4 species that definitely predominated our collection. We had Culex portici, Cockleitidia venezuelensis, Culex pedroi, and Aedes serratus. What I really want to draw your attention to is this species here, Culex portici. So we screen these mosquitoes by homogenizing these little mosquitoes up and then using the supernatants to infect cell cultures. And then if there's viruses present, the viruses will kill the cells and you can visualize the cell death and know that there may be a virus present or not. We isolated 85 cytopathic viruses. So these are viruses that kill vertebrate cells. 47 of which belong to one genus, the alpha viruses, and the others belong to the genus orthobunioviruses. If, however, you take a look at that species that I mentioned before, Culex portici, 45 of the 47 of the alpha viruses was from portici, and portici represented all of the Bemiti, all 17, as well as 13 out of the 15 or 16 caraparoviruses, a really high infection rate for these mosquito species. So just to summarize a little bit of the surveillance that we done in Trinidad, we isolated about 32 known vectors. Among our positives, we isolated 7 viruses from 7 vectors within our collection. 82 out of the 85 mosquito species, or 82 out of the 85 isolates came from melanoconian mosquitoes, and 78 came from one single species. So this likely represents our highest risk of viral infection if you're exploring certain habitats. So we had a high rates of both alpha viruses and orthobunioviruses, and all of which are known to be pathogenic in humans. So my lab is actually continuing these studies here in Virginia, where we're looking at avian and mosquito surveillance. And if there are any undergrads in the room and you're interested in doing some surveillance work, you're more than welcome to shoot me an email. Upon completion of my PhD, I moved to the University of Texas Medical Branch in Galveston, Texas. And there I encountered 2 people that really directed and really supported my career. The first was my postdoc mentor, Dr. Scott Weaver. So he recruited me directly from my PhD, and I spent 4 really, really good years with Scott. The other mentor was Dr. Bob Tesh. So those of you that may not know these 2 names, these are actually some extremely very famous people in my field. And Dr. Bob Tesh, who had nothing to do with my recruitment, really took an interest in me, and he provided a lot of support with respect to life as well as respect to arbovirus research. But at UTMB, I benefited from almost unlimited funding. So I did a lot more arbovirus surveillance work. I did some virus evolutionary work, looking at spatiotemporal spread of viruses. And that's where I started my vaccine development work. So I'm going to tell you a little bit more, and a little more detail about some of the vaccine development work that we're doing here at Virginia Tech. As I mentioned, we've developed vaccines for alpha viruses like chikungunya, it's a mosquito-borne viruses, and we've developed vaccines for flaviviruses, primarily Zika virus. We've also done some SARS-CoV-2 vaccine development. So I'll tell you a little bit more about our Zika virus work. So for those of you that don't know, Zika virus is a very important mosquito-borne virus. It was first isolated in 1947 in Uganda, and previously it was only known to cause sporadic outbreaks in Africa and Asia. But in 2014, it emerged in Brazil, where it rapidly spread throughout South and Central America. Really interesting thing about Zika virus is that it's not only spread by the bite of an infected mosquito, but it can be spread through sexual transmission. It can be spread from during pregnancy from mother to offspring, and it can be spread by blood transfusions. Zika virus disease is actually most often mild, causing things like headaches and conjunctivitis. However, infection in pregnant women is where the real risk of severe disease exists. And congenital Zika syndrome is a pattern of birth defects associated with infection of pregnant women. This is where there's congenital malformations of the fetus, the most famous of which is microcephaly. This is a narrowing of the head circumference, which results in developmental disabilities. So Zika virus is a classical arbovirus. As mentioned before, it's spread by infected mosquitoes to susceptible vertebrates. And these are the ones that everybody knows. It causes outbreaks in humans and animals. However, there's another group of viruses that infect only arthropods. It only infects the insects and cannot be spread to vertebrate animals. Fortunate enough for us, we were able to isolate one of these insect-specific viruses. In this case, it was an insect-specific flavivirus, so it's closely related to Zika. And we isolated this virus from Trinidad, from some of our mosquitoes in Trinidad. This virus, just like what we expected of an insect virus, it could not cause any cytopathic effects. It didn't kill vertebrate cells. It couldn't grow in vertebrate cells. But when we sequenced it, we can see that it actually does look like it is closely related evolutionarily to the vertebrate pathogenic flaviviruses, as you can see here. You see a retrovirus here. One unique trait associated with a retrovirus is that a retrovirus was the first virus that we were able to show that actually enters vertebrate cells by a receptor-mediated endocytosis. So we were pretty happy. We found a virus. We were interested in making we found an insect-specific virus. We were interested in making a vaccine. So we developed a vaccine by creating a chimeric virus. And to do this, we used our retrovirus that we isolated, and we substituted the structural proteins, or the pre-M and E proteins of Zika, into the retrovirus genome. So now we have a virus that looks like Zika on the outside, but the rest of the genome is replication deficient in vertebrate cells. So it can't replicate in a vertebrate, but it looks like Zika on the outside. So this virus grows well in mosquito cells, so it makes it very easy to produce and at very high titers. You can administer very high quantities of this vaccine to a person, and it presents these antigenic proteins in its native conformation. So it's very highly immunogenic. It produces a really strong immune response. Even though it's very safe, it can never replicate in a vertebrate. So we showed that very quickly. We did a very simple experiment. I'm not going to go into too much detail, but we would infect vertebrate cells in cell culture and look for replication of the virus. Here you can see a Zika in green and the Aripo backbone in blue. And you can see that in vertebrate cells, there's almost never any replication. It just tapers off over time as the virus degrades over time. If you look at protein expression, you also see that we never detect a Zika protein expression in any of the vertebrate cells we test, only in the mosquito cells that we test. So this sort of suggests, yes, the virus is not replicating in these cells. The virus is also very safe. Even when we use the most permissive environment that we have and we administer the virus directly to the brains of mice, we see that these mice continue to gain weight normally. Zika is in green, they gain weight normally, but Zika infected mice, they lose weight and they die within 7 days of infection. And if you dissect out these brains, you can actually see that Zika brains looks completely healthy in contrast to Zika brains, where there's a lot of neuronal necrosis and hemorrhaging. So we did find a very safe vaccine. So what we wanted to do is we wanted to see, is it actually an effective vaccine? And to do this, we set up a short-term vaccine efficacy study. So I just want to briefly go over this with you so you can see sort of how we test vaccines in the lab at this stage during preclinical development. So we would vaccinate our mice on day 0, and we would monitor these mice to see if they present any adverse effects to the vaccine. We'd also take blood samples to look at the immune responses over a month. After that month passes, we would challenge with a lethal dose of Zika virus to see if the vaccine can protect against the challenge of virus. After challenge, we would take some blood samples to see if there's any Zika virus present in the blood, and we would monitor these mice to see if they lose any weight or if they die because of the Zika virus infection. So, Zika, that's our experimental vaccine, is shown in green, right? And our sham, which is mice that was vaccinated with PBS, or control diluent, and then challenged with Zika. So those are the two main groups I'm going to draw your attention to today. So after we vaccinated these mice and we challenged with Zika, you can see that in green, in all cases, we see no death of these mice or no weight loss. In contrast to the sham mice, where we see death at 7 days post-infection and significant weight loss after Zika virus challenge. So this is pretty good evidence that the vaccine is protecting these mice from Zika virus disease. When you look at virus in the blood, you can see that the vaccine also completely prevents the presence of Zika virus in the blood after challenge, in contrast to the backbone and the sham, where you see lots of virus in the blood. And this makes perfect sense to us because upon looking at our immune responses, we look at antibody-based responses here in this case, you can see that Zika induces high, what we call neutralizing antibody responses. And these are antibodies that can neutralize the virus, sort of like to stop the virus from infecting cells. And you can see it's as early as 7 days post-vaccination in this case, and it goes up, it elevates to about almost 3 logs by 28 days post-vaccination. So our vaccine was safe. It seemed to be very effective. So the next thing we wanted to look at was, can it do what we really needed to do? Stop it from prevent these congenital malformations. So luckily for us, there's an animal model available for that. So we did a very quick study where we vaccinated some mice. We would then mate these mice a month later. Once we detect a vaginal plug, which means that the mice are now pregnant, we will call that an embryonic day 0.5. So pretty much 10 days after they become pregnant, we would challenge them with Zika, and then we would monitor them for weight loss and death. So what our data shows, our Zika, again, always in green. You can see that the dams, the moms, they never lost weight in contrast to the sham controls. The moms presented, the dams from sham vaccinated mice presented significant viremia, but our Zika completely protected the moms. Now, when we start taking a closer look at the pups, so these are the neonates, you can see that our Zika vaccinated moms and their neonates showed no significant differences in weights between the pups and the unchallenged pups. However, the sham vaccinated Zika challenged pups, they had significant weight loss. And if you look at the maternal placenta, maternal spleen, again, our Zika completely protected these, as well as the embryonic brain and the maternal brain samples. So this is exactly what we were looking for. So I just want to summarize that one really quickly. So we had a safe vaccine. It maintained its natural host restriction, which means it couldn't replicate in vertebrate cells. It was highly immunogenic, produced strong antibody responses as early as 7 days. We showed strong cellular immune responses, which I didn't show the data for here. And it completely protects immune-competent, immune-compromised mouse models from Zika virus disease. And it completely protected against in utero Zika transmission. After finishing my postdoc, I came over to Virginia Tech. And here, I was fortunate enough to find 2 really important mentors in my life, Dr. Timothy Kring from the Department of Entomology and Dr. XJ Meng at the vet school. Under their support and leadership and guidance, I do pursue their direction a lot of times. I have been a relatively successful assistant professor, if you like to call that. A lot of times the metric for success as an assistant professor is funding. I don't really necessarily believe that at all, but I just wanted to show you. So I have a very diverse research program, primarily driven by my very enthusiastic and motivated students. I also have been able to be fortunate enough to receive a lot of funding from multiple agencies like NIH, HHMI, and some local Virginia funding. But even more important than funding to me is the impact that we can have training the next generation of scientists. So I wanted to go through and say from right to left here, I guess. I'm going to introduce you very quickly to my very, very nice team. And I'm very proud of our graduate students. Ms. Manette Tanalas on the right. She's a Carver Fellow. Danielle Poirier is my most senior graduate student and graduating next year. William Stone, who is on an NIH GPP fellowship, and Crisangel Lopez, who is on an HHMI Gilliam fellowship. So these are my current students. And previously, we've had research opportunities, more than 16 undergraduate researchers. We've hosted more than 4 vet students, 4 medical students, and we graduated 2 master's students. And I think this is more of a compelling story as to how I measure success of an assistant professor in general. So what should you do for the next generation? And that's what I really want to talk to you guys a little bit about. Again, though, for anybody in the room, if you're interested in any of the ecology work, any of the vaccine development work, what we're missing from our team right now, though, is you. So please feel free to email me. I'll be happy to communicate more with you. So I just want to talk a little bit more about you guys in the audience. You know, when we start our academic career, or when we start our career in general, when we're pursuing our bachelor's, we see the road as one long road where we put in our time, we put in our effort, we put in our hard work, And eventually we get to the end and we make bank, because financial security is one of our driving factors. That's our expectation. Reality is there's going to be a lot of winding roads that you're going to have to navigate and stay on that correct path. But what happens now when you hit this wall? Because you're always going to hit walls. The recurring story that I wanted to bring up with every place that I've ever been to is the support you receive. You need to surround yourself with that support network that will push you further. And I don't only mean friends. I mean, I want you to reach out to the faculty. I want you to reach out to people senior than you, maybe even junior than you, but you have to surround yourself with those that will help you achieve excellence. And that's the most important thing. I think that will help you in many different ways. As I mentioned before, I wouldn't be in graduate school if I didn't get a letter from a faculty member. And that's really important. So when you do surround yourself with people, you do put in that hard work, that hard effort, and you do make it, then what's next? All right. Then I'm going to encourage you guys now to think about giving back to the community and giving back, and how can you support the next generation of scientists? So one thing that always comes to my mind, and one thing that I think about a lot, is why are students now so reluctant to stay in academia? There's always a few, very reasonable explanations. It's a lot of work might be involved. It's quite a stressful environment. Some people think there's no work-life balance. Some people would just like to relax and enjoy it when they're making bank. That's fair, right? Some people are afraid that they won't be productive enough to actually pay salaries and have to fire people. But I want to remind you about a few things and offer some encouragement about that as well. So I'm sure, since this is seed, I'm sure that you guys are probably well aware of sort of these statistics, all right? This was some statistics from the Association for Women in Science, where they broke it down by ethnicity, the distribution of diversity as you progress through an academic career. And as you can see, in gray, we see Caucasian population and people of color in orange. And as you can see, as you go further from your bachelor's to deans, you can see how this population sort of really decreases for people of color. And that's what we need to, that's what we need to address. When you drop out of this process, the impact that you have, you will not have an impact. And that's what we need. We really do need to have an impact. We need to stay all the way through. We need this bar to actually expand a lot more. One of the main reasons why people are always worried about it, about being in academia and staying in academia, is the financial support and whether or not you can acquire that financial support in academia. And that makes sense. If you looked at the data just prior to 2010, if you look at a general census, this is what a normal population of people looks like. This is Caucasians, Black and African Americans in purple. And the other population I always look out for is American Indians in gray. Right? If you look now at the faculty at medical schools, you can see it's about 3% Black, 0.1% Native American. And if you look at NIH-funded work prior to 2010, and you look at the African American, you see it's 1%, 0.4% for American Indians. Such low rates has definitely been discouraging. And I can see why that would induce, some sort of stressful state prior to accepting that. But I'm here to offer a little bit of good news. In 2010, 2011, a lot of funding agencies actually realized what they were doing wasn't working the best for everyone. Since then, they put a lot of programs into place that can now help accelerate the success rate for minorities in this case at NIH. And you can see that we now in 2020, 2022, we are a very high success in funding rate among minority populations. So that's one issue, assuming we're writing, same quality grants. It's one issue that we don't have to worry about as much, which is usually the major issue. So what can you do though? Why do we still want to pursue this? Now let's think about numbers. Let's just think about numbers. You are one person. I am one person. Let's say I have 200 trainees in total. all right? Your trainees are always going to reflect you. So you have 200 trainees. Let's say 100 of these trainees now stay in academia, and those 100 train another 100. You would then have in your generation, before you die, God forbidding any accidents, you could likely have 10,000 trainees that reflect your goals, reflect your thought processes, your expectations. And the next generation of that can have close to 1 million trainees if these 10,000 are successful enough to have another 100. And that's sort of the impact that one person can have, because it's very difficult to see or accept that one person can make a difference. So I just want to show you some very general numbers. And these are not numbers that's impossible. I've definitely seen labs where there's 100 people within like 4 or 5 years. So hopefully I've convinced you two guys of two things. First is build a strong support network, and please include faculty. We are definitely here to help. We're here to help with your research, with your advising. We are here to help. Just include us when you can. Make sure that we are invested in your success. That's just going to help you a lot in the long run. When you do become the master of your field, then come back and think, how now can I provide opportunities for others? Whether or not you stay in academia, even if you go to industry, try to give back a little bit once you do hit that pinnacle. Remember, you did have that support, and then you want to also provide that support. Now, if you're still having doubts a little bit, it's usually one reason that there's a lot of doubts in the mind of whether or not somebody can stay in academia, whether or not somebody should be a professor. So I have a quick little activity for everyone. All right. And I hope everybody that's on the audience can participate. Please do. What I'm going to do is I'm going to ask a series of questions. And for each question, I just want you to take one hand. If the answer is no to that question, I want you to take one hand and just put it across your chest. If you have another no, just take the other hand and put it across the other chest. Don't care about the people in the room. Don't care about the people next to you. If you're in a bus, it doesn't matter. If you're in a classroom, it doesn't matter. Just try to participate in the activity. Let that all just dissolve away. So first question. Have you ever been taught? What is it? Have you ever been taught or had a research experience with a Caucasian professor? If the answer is no, again, put a hand to your chest. Same question. Have you ever been taught or had a research experience with an African American professor? If not, put a hand to your chest. Have you ever been taught or had a research experience with a Native American professor? Again, if not, put the other hand to your chest. Have you ever been taught or had a research experience with an LGBTQ professor? Last question. Have you ever been taught or had a research experience with a Latinx professor? Now, if you identify with any of these folks, I want you to keep those two hands there. And I just want you to give yourself a nice little squeeze, give yourself a little hug and say that you can do it. Because odds are, if you've never had the opportunity and you interact and you identify as one of these people, is you might be inclined to think that you can do it because you've never seen it. But that's not true. Just want to encourage you guys, definitely, you can do it. So I just like to thank a few folks, my family. I grew up in a really nice, loving household. My mom, Deborah Auguste, and my dad, Albert Auguste, and my brother. My wife, Dawn Auguste, as you can see here, she's actually my lab manager in the lab. And some of my collaborators and mentors throughout my time, as well as some of the funding that I've received that has supported my research, as well as my diversity and inclusion efforts here at Virginia Tech. And with that, I'd like to say thank you, and I'll be happy to take any questions.
Thank you, Dr. Jonathan, for your great presentation. And I got a lot of questions for you. However, I'm gonna save my questions to see if there are any questions in the chat box. Students, participants, if you have any questions, feel free to put your question in the chat box, and we're gonna explore those questions with Jonathan and go from there. I do see one question that's in the chat box. If I were to start an academic lab, do I reserve the right to recruit undergraduate and master's degree students only? As an inspired professor, I might not feel prepared to take on a PhD students financially, mentally, and emotionally. I would want to focus on getting underrepresented students ready to take on the pearls of doctoral and postdoctoral I experience.
That's a great question, Martha. All right, I'll stay here. So that's a great question. So got good news for you a little bit too. And I see your point. So you may not feel like you, as you said, financially or mentally or emotionally commit to as long as a student, as a PhD student. There are institutions that would primarily, there are non-PhD degree granting institutions that have strong research groups. So for example, we just hired somebody onto our department who at their highest level only graduated master's students, never had to take on PhD students. This was not an R1 institution because they usually sort of focus a little bit more on the PhD levels. But there are opportunities at these institutions. Same, you can do the same degree of research. If you're doing excellent enough, if you're doing good enough research, you can maintain that level of funding and you can fund these master's students. So that is one way you can take things. The other way to take things now, or think about things, is 2 years is a really short snapshot. That is a very short space of time to really, really not only make an impact, but to have that student reflect who you may be or who you think that student can be if you want that student to excel in, to be themselves as they grow. And 2 years is very short. You would be surprised to know that 2 years, including courses, it's very difficult to do a lot. If you think about it like that way, and then you offer yourself the opportunity to do a PhD program of 4 years, you have a lot more time to develop students and really make them highly competitive and really sort of like guide and support them throughout that time. But again, back to the original question, it is possible. Some institutions will be better receive that, recruiting only undergrads and masters than PhDs, but that is there, you do have that opportunity, depending on which institution you choose. Did I miss anything?
No.
Okay.
Now, at the beginning of your presentation, you indicated you had a 2.9. Did you ever face the imposter syndrome? And if so, how did you deal with that throughout your career? Because you identified 2 faculty members were superstars in their fields. And I don't know if that made you feel some kind of way or not. Would you speak to that?
Yeah, that's a great question. And I can probably tell you that everyone in this room would feel imposter syndrome at some point, especially as you advance throughout your career. I've definitely been guilty of that. I still feel imposter syndrome quite a bit. I don't think that might go away anytime soon. I think if you're able to, which is difficult, and it's going to be even more difficult for women, it would be if you can actually sit and objectively assess your career without actually criticizing yourself or knowing that or thinking you could have done things better. I think you might be able to sit and see, oh, I'm doing okay. I'm doing good. You might, be as, okay, let me phrase it like this. So as Tremaine mentioned, I had 2 real superstars. If I compare myself to them every day, I will always fall short. These guys have 400 or 500 publications. Take it, they're 70 years old in some cases. But you will fall short like that. But if you can objectively review yourself and say, I've done the best I can, and it's been and worked out really well, you really don't have to, you shouldn't suffer through that imposter syndrome, which unfortunately most of us, we almost always do.
Are there any other questions from the audience? If not, I have plenty.
Please feel free to unmute yourself and ask questions or put it in the chat. Definitely
Absolutely.
interested in hearing your thoughts as well.
Hello, my name is Joseph Sturgis. I had a question in terms of, I know in academia, getting funding and grant writing is a huge part of it. So my question is, how did you learn about writing grants or acquiring grants? And then how did you build that skill set?
Yeah, it's a great question. This is what it almost always comes back down to. All right, so I do, luckily enough, because this is actually what I present on most. There are so much more opportunities than you would think you had when you do become a faculty member. It first starts, and this is why you guys actually do need to pay attention to it when you do it. It starts in your graduate when you're doing your PhD. That little proposal that you're just trying to get done to be able to submit to get, become a candidate. It actually does start there. If you take it seriously, you learn enough in that initial portion just to be able to, okay, I know what a grant's supposed to look like. When you do a postdoc, or if you don't do a postdoc, in that case, we can skip that one, but if you do a postdoc, you usually get more opportunities to write grants. It all comes back to your professor and how much they can introduce you to it. In my lab, it's very unique. I have my graduate students and postdocs, they write lots of grants, and that's why they have so many different fellowships. So I introduced them to that very early on, and I introduced them to the hard ones, because once you can write the hard one, you get the easy ones much easier. Then that's the second step. But when if you even skipped a postdoc and you wrote one proposal as a PhD student and you make it to a faculty, seek the support of the institution. At Virginia Tech, for example, we have grant writing support within the department. We have grant writing support within the college. We have grant writing support at the institutional level. If my writing's horrible and that support doesn't help, then you depend on your colleagues. You can, you support your colleagues, you review theirs, they review yours, and you ask your colleagues then to read your grants and tell you what they think about it. And this is very important. Just the peer review, people that's in your field and not in your field, it's going to go a long way with the actual grants. So then, and in addition to that, there are agencies you can hire, for example, or get red tape review of a grant. You can send it to somebody and pay them money, and then have them review your grant and send it back, and you'll get your comments to address. There's so much effort into that, because once you bring money, the university gets money, the institution gets money. So there's always going to be a lot of support for getting grants. So that's one of the things I would probably say. Just get the experience you can while you can, but you will get the help on that front as you move forward. And make sure you find your mentors. They'll review them. They'll help you get those grants. I got my first grant after my 2 mentors reviewed it. I got nothing before that for 2 years.
Jonathan, do you?
Go ahead.
Jonathan, do you encourage students to bring to you ideas of fellowships that they would like to apply for?
Absolutely. I don't even I think my students would do that anyway. Yeah, so I encourage them to bring it forward. And in some cases, they write most of it. I just edit at the end. And in other cases, I have to do a lot of editing. Yeah, Secret Angel posted in the chat. Yeah, so definitely, I would definitely encourage students always bring things forward as they need to. As long as it's not going to completely take, some students will commit all the time to writing. As long as you don't then decide I want to write a grant and stay home for 2 months, because then if you don't advance your PhD, that might be in jeopardy of graduating, So that's a big call there.
We do have another question from Erica Cooper. Can you speak more about the importance, in your opinion, of the postdoc experience? What are the benefits of completing a postdoc before accepting a faculty position?
Hey, that's a great question, Erica. That's a great question. So each field of study is very different. I know a lot of PhD students upon graduation accepted faculty positions in economics and math. I've seen that happen a lot of times. In certain biomedical fields, the postdoctoral experience is, we call it almost like think of it like an add-on experience to the PhD. In the PhD, you had a lot more mentorship, or you had a lot more direction as a student. As a postdoc, you become much more independent. So as a postdoc, as an independent scientist in the lab, you then start mentoring students, which is number one, because even if you did that as a PhD student, now you're pretty much doing it independently as a postdoc. You can start writing grants, much more so as a postdoc, and grants that you, not only fellowships, but grants that you can get as a postdoc and be a research grant. As a postdoc, you also have opportunities to do postdoc to professor grants as well. It's called career development grants. So that gives you a little bit of opportunities there. But I think it's because of the newly found independence as a postdoc, because you might be very, very independent, depending on your PI. And the additional experience you get. So you add to your skill set. I guess that's the last one. So you might want to do something completely different than what you did in your PhD. Your postdoc's the time for that. You go, you learn something extra, you learn something new, something that you can merge with what you did during your PhD. So if, however, you did all of that in your PhD, let's say you did a 10-year PhD, for example, and you did 4 different projects, you wrote grants, and you don't need a postdoc, odds are. I've definitely seen it happen. I've seen it be successful. I've seen it successful in biomedical fields as well. It's just that it's an extra period of training, and it's an extra period of training where you might have more mentors that has different philosophies than what you might have had as a PhD. So I would say usually recommended in my field. In fact, it's almost required in my field. I wouldn't even seen it done once, but I just think of it as more, potentially more training and a way to enhance your skills and your CV.
Excellent. Are there any other lingering questions for Jonathan? This is a great discussion. If not, we can pause there. And Jonathan, I would like to say thank you so much for volunteering to do this presentation. I learned a lot, and I will definitely be reaching out to ask you more questions as I'm learning what students are needing, especially in the College of Engineering. Again, we appreciate all that you do for our students here at Virginia Tech. And again, thank you so much.
Thank you, Trey.